everybody, and welcome to today's webinar. My name is Melissa, and I'm a Scientific Training Officer in the EBI training team. In today's webinar, we're joined by Rosanna Saru, who is going to tell us more about the Enzyme Portal. And with that, I'm going to hand over to her to lead you through the webinar.
So good afternoon, everyone, and welcome to this webinar on Enzyme Portal. In this webinar, I'm going to do a practical walkthrough of the Enzyme Portal website. I will show you what type of information specific for enzyme can be found in it and how to search for it. But first, I would like to start by giving you a brief overview on how the Enzyme Portal came to be. So enzymes play essential roles in all biological processes and therefore have been increasingly important for various areas of research, such as medicine, biotechnology, bioprocessing, and drug design. Despite attracting a huge interest, enzyme-related information can be sometimes very difficult to find, as it's often spread across various resources, which often focus only on a specific aspect of an enzyme. For example, the PDB database provides mainly information about the 3D structure of a protein, whereas in a resource like Reactome, you will find information related mainly to the pathway where the enzyme acts. This can make finding enzyme-related information time-consuming, and also it increases the risk of missing important information. So the Enzyme Portal was designed to help solve this problem by bringing together in one place enzyme-related information extracted from various resources. So you have among these resources UniProt, which is a protein sequence and function database, the RIA database, which provide biochemical reaction, Intents, which provides nomenclature and classification of enzyme catalyse reaction, PDB, which I mentioned earlier, which provide the 3D structure information, CHEMBL, a database of bioactive molecules with drug-like properties, Reactome, a pathway database, MCSA, which provide information about reaction mechanism, KB, a small molecule database, and OMIM, which provide information for human genes and genetic disorders and traits. And finally, Europe PMC, which contain all the scientific literature. From all these 10 resources, the portal extract and display information regarding the function of the reaction, the mechanism, the pathway, and so on, making it easily available for the user. So this is the Enzyme Portal homepage. It is divided into 3 sections, which I will explain in more detail in a moment. But briefly, at the top, you have a search box, which allows you to perform general searches. In the Browse by Enzyme section, you can make your search more specific by searching enzyme according to their classification, disease, taxonomy, pathway, enzyme families, or cofactors. The last section of the homepage provides you with tools to analyze data. Let's just start with the general search box at the top. Here you can query the portal using keywords such as enzyme name, gene name, or substrate. And for those of you who are familiar with them, you can also use UniProt accession numbers or KB or RIA identifiers for compound and reaction. Below the search box, we have provided some example of what type of search you can perform. To show how this works, let me use an example. So let's just say that you are interested in enzymes that are involved in the metabolism of catecholamines, which are a group of neurotransmitters which contain a catechol group. As I start typing in the box, an autocomplete menu appears, which will provide me with some suggestions that could match my query. As catecholamines have a catechol group, I decide to select catechol to keep my search broad. This will launch the search and will take you to the enzyme result page where the enzymes that match the query are displayed. As you can see here, there are 44 results for the catechol search. In this page, all the protein hits are grouped according to their catalytic activity, which is represented by the 4-digit enzyme classification number, commonly known as the EC number. On the left of the page, you can find filters that you can use to narrow down the search to a specific class of enzymes. For example, you could restrict your search to enzymes that have transferase activity. To view all the proteins that are associated with one of the enzyme activity, you just click on the name, and this will expand the menu with several of the protein hits. But as you can see, we only display up to 7 proteins per enzyme activity. And to access the full set of the associated protein, you need to click on the View All 17 protein. This will open the protein results page where all the 17 proteins matching your query that have actual catechol methyltransferase activity are listed. So for each protein, we provide a name which comes from UniProt, a summary which includes, if available, the protein function, the EC number and its associated catalytic activity, any associated disease, and we also provide the 3D structure if it's available. As you can see here, there is also a section about species. To facilitate the display of the results, all the orthologs for a given protein are grouped into one entry. And here we list all the species where a similar protein has been identified. And I will come back to this later. In the protein page, you have also the possibility to store a selected protein into the basket, so you can come back later to look at them. On the left-hand side of the page, you have additional filter that will allow you to narrow down the search to a specific organism, enzyme class, cofactor, inhibitors, or disease. For example, let's just say you would like to restrict your initial search to retrieve only human enzymes that are involved in schizophrenia. To do this, you select human in the organism filter and then schizophrenia in the disease filter. This will update automatically the enzyme result page to display only the enzymes that match your criteria. And here it is. So in this case, there is only one protein that matched the criteria. If you would like to see more information about this protein, you just click on the protein name, and this will take you to the protein page. This page displays various information about the protein that you can directly access using the menu on the left. At the top of the page, you have also an organism box. And as I mentioned earlier, all the orthologs are grouped into one protein entry. This box will allow you to see the information related to a specific organism. For this, you just select the organism in the drop-down menu, and the protein information for this species will be automatically displayed. So let's explore the protein page in more detail. So in the protein summary section, you can find functional information, which has been extracted from the UniProt database. You will also have a direct link to the UniProt protein page, where you can explore further other protein features. We also provide the UniProt ProtVista sequence viewer that will allow you to see sequence-related features in a graphical view. For instance, you can see where the catalytic sites are or if any variants exist. The protein structure section provides the 3D structure from the PDB resources if it's available, as well as a direct link to the corresponding entry in PDB. The reaction and mechanism section displays the enzymatic reaction catalyzed by the protein with the direct link to the REA website and the reaction mechanism from MCSA if it's available. But as you can see here, there is no reaction mechanism for the human methyl transferase. But if you use the organism box that I mentioned earlier at the top, you can find out if the mechanism has been described for another species. And indeed, if you select the rat organism, a reaction mechanism is available with its link to MCSA. The pathway section provides a list of the biochemical pathway which the protein is involved in. This information comes from Reactome, and a direct link is also provided to allow you to explore the pathway in the Reactome database. The small molecule section provides information about cofactors. Here you can also find known inhibitors and or activators from the CHEMBL resource. For example, catechol, for the catechol O-methyl transferase, there are 2 known inhibitors. To get more information, a direct link will take you to the compound page in CHEMBL. The disease section provides a description of the related disease from the OMIM resource, and there is also a link to UniProt where additional information about variants, for instance, is available. Finally, in the literature section, you will find all the papers related to the protein with the link to Europe PMC. Beside the general search box, the website provides an alternative way to query the portal using the Browse Enzymes tab. This will allow you to make your search more specific by browsing enzyme associated with a specific enzyme classification, a specific disease, species, pathway, enzyme family, or cofactor. To browse by enzyme classification will allow you to search enzyme using the hierarchical classification of the EC number provided by the Intense database. For example, let's say that you are interested in finding protein phosphatase, which are known to hydrolyze phosphoric monoester bonds. Based on the EC classification, I will start the search by selecting hydrolases, then hydrolases which act on ester bonds, and more specifically on phosphoric monoester bond. At the end, you will get a list of various protein phosphatase candidates. If you click on one of them, it will take you to the enzyme page. This page contains various information about the enzymatic activity. Here you can find information on the enzyme names, the reaction participant, if the enzyme requires cofactor, and more importantly, a list of the protein which have this specific catalytic activity. To view the full set of protein, you just click on the View All button, which will display them in the protein result page that I described earlier. In the page, you will also find information about the reaction mechanism if available, and also paper that discuss this enzymatic activity. If you are less familiar with the EC classification, you can also browse the enzyme by families. For example, you can search enzymes that belong to the MAP kinase or methyl transferase families. The list of these families comes from UniProt, and they are arranged in alphabetical order. The page, as you can see, displays only a limited list. So to search for family that are not displayed on the page, you can use the search box on the top of the page. This search box uses a control vocabulary, which means that as you start typing the first few letters of a family name, like I did here, a drop-down menu will appear where we can select the most relevant suggestion. The browse by disease allows you to find enzymes that are associated with a specific disease. The disease list comes from the UMIM database. As for the enzyme family, the diseases are arranged in alphabetical order, but you can also use the search box, which has auto-complete functionalities to find your disease of interest. For example, let's say you're interested in finding protein associated with schizophrenia. As you start typing, the drop-down menu will appear with several suggestions, including schizophrenia. Once you have selected it, it will take you to the enzyme result page where the hits are displayed. And as you can see here, for this disease, there are 4 enzyme activity that have been found, including the one mentioned earlier, the catechol methyltransferase. The browse by pathway will allow you to find enzymes that are associated with a specific pathway. The pathway list again comes from the Reactome database, and similarly to the disease, they are arranged in alphabetical order and they can be searched via the search box at the top. The browse by cofactors allows you to find enzymes that require a specific cofactor for the catalytic activity. The cofactor list comes from the UniProt database, and again, like the disease and the pathway browse by section, they are arranged in alphabetical order and they can be searched via the search box at the top. The last specific search that you can do in the Enzyme Portal is the one that allows you to browse enzyme by taxonomy. This will allow you to retrieve enzymes from a specific for a specific species. In the search box at the top, you can use either the common or the Latin species name. For instance, human or Homo sapiens will work for the search. We also provide a direct access to well-characterized species, especially those that are used as model organisms, such as mouse, E. coli, or C. elegans. Besides the 2 types of search that I just described, the Enzyme Portal provides also tools to analyze data. The Transform Miner tool was developed by Janet Thornton Group at the EBI, and this tool allows you to find enzymes that act on a specific molecules or enzyme that could catalyze the transformation of a given substrate into a given product. If you are interested in this tool, you can find more information about it on the Transform Miner website. The Search Sequence tool allows you to find enzymes that are similar to a particular protein sequence. It uses the NCBI BLAST, and to use this tool, you paste your protein sequence in the box and it will return a list of similar protein in the Enzyme Portal. The last tool is the Compare tool, which will allow you to compare the feature of 2 enzymes. For instance, you would be able to compare protein sequences or their involvement in biological pathway. For this, you just select your enzymes of interest and add them to your basket, and then you click on the Compare tool. So I hope this overview of how to find enzyme data in Enzyme Portal has been useful. You can find additional help on the website in the FAQ pages, as well as more information about the various resources that the Enzyme Portal queries. If you have any question, don't hesitate to contact us through the feedback page that can be accessed at the top of the home page. And also, we are conducting a survey to try to understand what type of enzyme-related information people are interested in. So this would help us to improve and develop new functionalities for the portal. So we would really appreciate if you could take a few minutes to complete it. So this is the end of this webinar. Thank you for listening, but we will stay online for a few more minutes to answer your question that you can type in the chat box.
Thank you, Rosanna. That was quite interesting. As Rosanna mentioned, we do have time for questions. If you do have questions, please do write them in the chat box, then we can read them out and answer them live. In the meantime, if you could just switch to that last slide for me.
Sorry.
Yep. So just a reminder that this webinar is part of our regular series of webinars that we run on Wednesday afternoons. You can find out more information about what's coming up in the series on the EBI training webinar pages, which you can see linked on this slide in front of you. There's also information on there about other webinars that are being run by the EBI and associated projects. Finally, a quick reminder to please do fill in the feedback survey that will launch when the webinar ends. So let's have a look and see if there are any questions coming through.
So there is a question about the subcellular location for an enzyme. So this kind of information can be found in a UniProt database. For the moment, we don't display it in the Enzyme Portal, but by clicking on the link for the UniProt protein page, you can see them. Someone is asking if the Enzyme Portal can be used for expression studies. So for the moment, we don't provide any link to any expression database, like for instance,
Expression Atlas?
Expression Atlas, sorry. But this is a feature that we can try to see if we can add it to the Enzyme Portal as well.
Okay, as there don't seem to be any other questions, we will wrap up the webinar there for today. Thank you very much, Rosanna, for presenting the webinar. And thank you very much to all of you for joining. I hope you enjoyed it and hope to see you again in another webinar.